Para last na matapos ako.
Ayusin na natin.
Okay, good day. So for our oral presentation, so we have our title, Comparative Evaluation of Enzymatic Activity of Plant and Animal Catalase as affected by temperature and pH. So this is a special research on science, technology, and society, and this is group 3 from the DVM 1A. So our members include CASTRO Andrei, CRUZ Prinz Laurence, David Tricia Mae, DIZON Angelyn, DIZON Sarah Joy, of course Trudy, LIZO Deniel, MANANSALA Michael, MARUYAMA Maria Nozomi, Martin Rain Kian, and Mendoza Ayla Graciela. So for the introduction, we have Miss Tricia. Go, Tricia.
Good day, everyone. I am Tricia Mae G, the one who will devise the introduction of this research. According to Matsala and Niemi in Biological Systems in 2009, enzymes are major catalysts. Catalyst is an enzyme that naturally found in almost all species. On the same year, Fastred said that the biological processes will occur at a much slower rate without enzymes, which will cause harmful effects in organisms, as food molecules will be broken down at a much slower pace in the absence of digestive enzymes in animals. And the need for energy and nutrients will not be given immediately, which is very important for the survival of animals. As what Schreiner et al. On 2005 and Mamgaya and Matson on 2015 stated, in an animal, the enzyme is mainly present and is presenting abundant in the liver, which acts to eliminate hydrogen peroxide that causes oxidative oxygen and water damage. In addition, this enzyme is also present in plants that operate with the enzyme. This research, which aimed primarily to demonstrate the mechanism of the scientific method, was therefore intended to examine the rate of reaction between animal and plant catalysts by comparing samples using available household materials to be addressed later by my fellow members. Thank you.
Good day, everyone. I'll be glad to discuss the statement of the problem. I am Andrei Castro. Now, we observe the enzyme activity based on or as affected by or kung ano yung dalawang factor na yun. Yung varying pH, which is the acidic and alkaline, and varying temperature, which is the hot room and cold temperature. So, next slide, please. So, ayun. Yung number 2 of the statement of the problem in this study is, we focus naman on the animal catalyst, same as enzymatic activity exempted by varying pH and varying temperature. And the last SOP is, so next please. Yan, so since we are dealing with comparative analysis ng plant and animal of catalase, for the statement number 3, we are to compare the plant and animal catalase in their enzymatic activity as affected by the same factor, yung pH, acidic and alkaline, and the varying temperature, the hot room and cold temperature. Thank you.
Hi, good day. I am Angelyn T. I am the one who assigned hypothesis of the study. The following are the hypotheses of the study. The two are null hypothesis. Number 1, there is no enzyme activity observed in plant catalase as affected by varying pH and temperature. Number 2, there is no enzyme activity observed in animal catalase as affected by varying pH and temperature. Number 3, plant and animal catalase showed a difference in rate of reaction. Thank you.
For the methodology, good day. I am Ayla Graciela Mendoza, and I will be the one to present to you the methodology of our research. So for the methodology, we did an experimental type of research, so we used descriptive method. We use descriptive method since we did not use statistics. It will just become statistical experiment if we use ANOVA. Since we just use our site or we have just observed the bubble activity and the rate of reaction, so it is more appropriate to use descriptive method. The presence of the bubbling activity and the rate of reaction, we gathered this from these journals that are presented to make our method valid. We adopted our protocols from the Massachusetts Institute of Technology Laboratory Manual, biolibretext.org, and scientificamerican.com, which are educational organization website, and the journal Iwase et al. 2013. For the visual representation of catalytic activity, these representations will give everyone some information on what we have observed during the experimentation. If there's a bubble, that means there's the presence of enzymatic activity. The other one that does not have bubbles shows the absence of enzymatic activity. The bubbling activity corresponds to the equation where two molecules of H2O2 and then catalase acted on this hydrogen peroxide and it produced water na sumama po sa solution and then oxygen which is the indication of bubbling activity. This explanation is from Reigne K 2010 Catalase Test Protocol from American Society of Microbiology. For the rate of reaction, we just we just use a timer. We timed or we recorded the time of reaction when it started to bubble. In our method, we use hydrogen peroxide as a substrate, which means ito yung magre-react sa enzyme, which is the catalase. So yun po, tinimer po namin siya. Then the first bubble that we saw, that is when the bubbling activity started. We observe how fast or slow the bubbling activity is. For the bubbling activity, this is the representation of the bubbling activity. The first one shows no bubbling activity. The second and third one shows slow bubbling activity. And the last one has a rapid bubbling activity. So the bubbling activity is analyzed as to its descriptive kinetics because we relied on the visual representation of the activity that is happening. Thank you.
Good day, everyone. I am Prince Laurence Cruz, and so here are the materials that we use in this research. So for the wet samples materials, we use 2 small raw potatoes, 8 small slices of raw chicken liver or raw pork liver, 1 small bottle of agua oxinada or hydrogen peroxide, 1/4 cup of vinegar, 1/4 cup of alkaline water, 1 cup of tap water at room temperature, 1 cup ice cold water, boiling water, and distilled water. Next slide, please. And for the dry materials, we use timer, paper and pen for recording results, pair of tweezers, small colored balls, thermometer, and 1 medicine dropper. Thank you.
For the methodology, here are the treatments for pH and temperature. For pH, in T control, we have raw potato and raw liver only. For the T1, we have raw potato and raw liver plus vinegar plus agua oxinada. For T2, we have raw potato and raw liver plus alkaline plus agua oxinada. For the treatments for temperature, we have raw potato and raw liver only in T control. In T1, we have raw potato and raw liver plus room temperature plus agua oxinada. For T2, we have raw potato and raw liver plus hot or boiling temperature at 100 degrees Celsius plus agua oxinada. For T3, we have raw potato and raw liver plus cold temperature at 5 degrees Celsius plus agua oxinada. Here is the method flow chart per pH. We started by the samples, which are the raw potato and raw liver, where we cut them into 2 centimeter in length and 1 centimeter thick. Put slices in separately in 3 colored containers, then label the containers with T control, T1, and T2. Where in T control, we just put the raw liver and the raw potato only. And for the T1 and the T2, um, we leave them for 10 minutes soaked in vinegar. Then we added 10 drops of hydrogen peroxide. Then we started to time it and then observe the bubbling activity of these samples. And here is the method flowchart for temperature. Same samples, raw potato and raw liver. We did the same procedure, only we soak the raw potato and raw liver with tap water, cold water, and boiling water. Then we added hydrogen peroxide. Then we observe if there's bubbling activity. Thank you.
So, good day. I'm Kian Martin and I will discuss the results and discussion. So, we have the pH and temperature influence in plant enzyme catalase, and as you can see here, the table 1 shows the activity the plant's enzyme as affected by varying pH. The acidic solution used in the experimental in vinegar, which has a pH level at 2.5 to 3. T control shown inside of the table that plant enzyme did not produce any enzyme activity and bubbling activity, and it supports that Stern et al. And Gardner that the enzyme or protein molecules are easily chemical compound wandering inactive or lose their functionality. So T acidic. T acidic is the plant enzyme that acidic solution did not produce any enzyme activity and bubbling activity, and it supports that Gardner stem 2006 that acidity is the solution of enzyme inactive and able to catalyze. So T alkaline is plant enzyme with alkaline water. It did produce enzyme activity with bubbling activity. So next slide please. So table 1 2, the enzyme activity in plant catalase as affected by varying temperature. So T control, the plant did not produce enzyme activity and bubbling activity. And T room, which is produce 10 seconds enzyme activity and rapid rapid bubbling. T hot did not produce enzyme activity and bubbling activity. And T cold, it did produce enzyme activity and still produce in slow bubbling. And it supports all that according to Nelson and Gardner. This is due to the inactivation enzyme by heat, not its protein structure of catalase. Thank you.
Hello everyone, I am Michael Manansala and I will discuss our observation with the plant animal catalase treated in measuring acidity and temperature. The table that you can see on your screen right now aims to show how enzyme activity of animal catalase is affected by solutions with varying pH. For T control, we didn't do anything to the liver, therefore there is no observation obtained. T control is just used for comparison. In the acidic solution, there is no enzymic activity that happened. According to literature, acidic solution damages or disable protein molecules and catalase has a specific range of pH where they can function, which is 7 to 11. Therefore, the catalase of the liver is denatured by the acidic solution. However, alkaline solution didn't do anything to the catalase and it works perfectly with it. Next slide, please. This table aims to show how temperature affects the animal catalase. At room temperature, the catalase works perfectly and showed rapid bubbling for just 1.30 seconds after the first drop of hydrogen peroxide. However, in hot temperature, the catalase didn't yield any catabolic activity. Based on literature, the capacity of macromolecules or enzyme are affected by hot temperatures. Therefore, hot temperatures disabled or damaged the catalase. That's why it didn't show any bubbling activity. Meanwhile, the cold temperature also conveyed enzymic activity, but relatively slower than in the room temperature. 2 minutes and 20 seconds upon the first drop of hydrogen peroxide, bubbles started to appear. Therefore, cold temperature doesn't disable the enzyme, but it slows down its reaction. The next slide will be presented by another group member. Thank you.
For the last two tables for the results and discussion, I'm Sarah Joy Dizon, who will present the remaining results of the study. So for these two remaining tables, we actually focus on the comparison of the plant and animal catalase. So first table, table 3.1. So plant and animal catalase as affected by varying pH. So if we can see for the rate of reaction and bubbling activity in comparing the two samples, plant and animal samples. So plant in this case is raw potato, which is very rich in catalase enzyme, and animal sample, which is the raw liver, which is also a part in an animal that is very rich in catalase, since the liver is a detoxifying organ. And since it's a detoxifying organ, it actually prevents the body from oxidative damage. So catalase does the job for that. Okay. So for the treatment control, wherein there are only raw sample, so we didn't see any observation, since this is only a control um, treatment, and it's for comparison only. So the very interesting treatment in this particular table is for treatment 1 and treatment 2. So treatment 1 is treated with acidic solution. So acidic means it's very high in hydrogen ion, so acidic siya. So if we could still remember, acidic solutions have a pH level ranging from 1 to 6.5. So in that table, it did not produce any rate of reaction and bubbling activity. So therefore, based from literature, acidic solutions affect the structural integrity of molecules, especially proteins, since catalase is a protein. Next is treatment 2, treated with alkaline solution. So the alkaline solution in both plant and animal samples showed an enzymatic activities. So the rate of reaction, it's fairly similar with 1 second gap. And they also showed the same bubbling activity. So this means that the alkaline solution did not do anything to the structural integrity of catalase. And we know from literature, based from the presentation of other members, catalase works best at a particular optimum range, which is 7 to 11. Others or in other samples or other sources, 7 to 9, and it depends on the animal or plant sample. But in this case, if we are to generalize based from literature, catalase works best in that particular range, which is 7 to 11. Next slide, please. For the last slide, so if we analyze the varying pH in the previous slide, the last slide is for varying temperature in catalase. So if we observe the table for treatment control, no observation made, since it's just for comparison and we didn't do anything on that particular control treatment. So what is interesting in this particular table are the three treatments. So treatment 1, which is under room temperature, treatment 2, which is under boiling or hot temperature, and treatment 3, which is under cold temperature. So if we are to compare each of the three treatments, treatment 1 and 3 showed enzymatic activity, but they differed in the rate and bubbling activity. So napakabilis ng rate of reaction sa treatment 1, which is room temperature. Room temperature, that's just around 28 to 30 degrees Celsius. So in this case, it shows that the room temperature did not do anything to the capacity of the catalyst to react on the hydrogen peroxide, thus giving this particular bubbling activity na rapid. So mabilis yung bubble niya. The indication of the degradation of hydrogen peroxide into water and oxygen. And then treatment 2, which is under boiling and or hot temperature. So walang binigay na enzymatic activity, zero sa rate of reaction, and then no bubbling activity. So based from biochemistry books and organic chemistry books, including several journals regarding factors that affect enzymatic activity. So very high temperature affect the integrity of the structure of macromolecules, especially proteins, since they are very sensitive to changes in temperature. In this case, treatment to hot temperature, so it affected the very integrity of the structure of catalase, thus producing no enzymatic activity. And the last treatment, which is treatment 3, cold temperature, the same siya with treatment 1, nag-show siya ng enzymatic activity, but it is very slow. Slow yung reaction. So, nag-react yung catalase sa hydrogen peroxide at 50 seconds sa plant to 2 minutes and 50 seconds naman sa animal. And it showed for both samples low bubbling. So therefore, the low temperature slowed the reaction pero hindi niya dinamage or hindi niya dinestroyed yung structural integrity ng catalase. And that's it for the results and discussion. And next for the conclusions of the study.
I am Nozomi Maruyama from Group 3 and I'm here to present the first and second conclusion of our experiment. In this study, we have concluded that plant enzyme catalase found in raw potatoes showed different enzyme activity as affected by varying pH level and temperatures, while animal enzyme catalase found in the raw liver sample showed different enzyme activity as affected by varying pH level and temperatures. Briefly, both plant enzyme and animal enzyme catalase have different enzyme activity or reaction depending on the treatments made. The third conclusion and summary will be presented by the next presenter.
I am Deniel E from Group 3, and I'm here to present the third conclusion and summary of our experiment. We have concluded that plant and animal enzymes did not show a vast difference in enzyme activity as affected by varying pH and temperature. To summarize our research, we use the descriptive method, meaning we describe the result depending on our observation. This study primarily aimed at illustrating the process of the scientific method to analyze the rate of reaction between animals and plant catalase, using available household materials by comparing samples. In this study, potato for plant catalase and chicken liver for animal catalase were used. Catalase rate of reaction in different pH and temperature have different results. We have found out that there are various results depending on the treatment used.
And that's it. Thank you for listening for on our oral presentation. Okay na.